Good afternoon. Thank you for the introduction, and I'm sorry I can't be there in person. I hope this is the last time I give a virtual presentation, but I've said that before. Today, I'm going to share research that demonstrates management influences soil microbial communities and that these changes are associated with soil condition and crop performance. On the way, I will introduce a concept, emergent properties, and discuss this concept in relation to our current research. This is my previous ride, the Burb, War Wagon, Big Red with a hint of oxidation. By any name, this is a highly engineered mechanical system for personal transportation. The more gas I put into the tank, the further it goes. The more I press on the accelerator, the faster it goes, and the faster it uses gas. And with a 4-barrel carb and a 40-gallon tank, you can actually watch the fuel gauge drop at 80 miles per. This system has a predictable function and set of responses. There are no biological parts. In contrast, these cropping systems are full of biology. Plants, insects, microbes, animals, with a little bit of engineering. The annual performance of cropping systems, quantity and quality of crop or forage, is affected by a myriad of factors, including long and short-term management practices, the sun, temperatures, specific management events during a season like tillage or spraying, pest or pathogen outbreaks, and of course, precipitation, how much and when. And also less frequent events like large storms, windstorms, rainstorms. Those don't occur on any particular schedule. But most of these factors appear on multiple timescales: daily, seasonal, annual. And therefore, the outcomes are not always predictable for this complex biological system due to interactions among numerous biotic and abiotic factors. Which brings us to emergent properties. Emergent properties are properties that are only evident when the entire system is examined. It cannot be deduced by study of parts or individual factors. And in particular, these last three, they are a consequence of multiple interactions. They are a hallmark of complex systems, and they are a hallmark of biological systems. Low input, diverse cropping systems may function in fundamentally different ways than high input, simplified systems. It has been shown that low input, diverse cropping systems have characteristic microbial community structure and function, which translates into more efficient carbon use, more relationships among each other, that is, the microbial groups in the soil and the insects, and of course also the plants, the relationship with plants. These properties of the soil microbial community are emergent in their own right, and together with other factors influence these important system-wide emergent properties such as soil carbon dynamics, nutrient fluxes, pathogens and pests, and crop performance. On to the research, where we will examine emergent properties in our own experimental cropping systems. We conducted a long-term study on the effects of corn residue removal and cover cropping in a no-till corn soybean rotation. This field experiment was established in 2000. We originally had three levels of corn residue removal. In the next two slides, I'm just going to use two. The top one was, of course, where we just removed grain and we left all the other corn residue on the surface of the no-till plots. And then for the treatment where we removed all the corn as silage and baled it and took it away. The fall cover crop treatments were installed in a split plot arrangement in 2005. This graph illustrates the interaction between the treatment factors, cover crop and residue management, on the erodible soil fraction, that is, aggregates less than 1 millimeter in diameter. These are the size of aggregates that are most easily eroded by wind and water. The amount of erodible fraction is normalized to 100% for the residue removed, no cover crop treatment. Higher erodible fraction is bad, lower is good. And as you can see, with the residue removed, the addition of cover crop lowers the erodible fraction of soil. However, when the residue is retained, the erodible fraction is already low, and the addition of a cover crop makes no additional effect on this measure. This is a similar graph looking at the same treatments and their interaction, but the measurement is soil microbial activity, which is again normalized to 100% for the residue removed, no cover crop treatment. In this slide, higher is better, lower is bad. And you can see with the residue removed, the addition of cover crops does not increase the microbial activities. However, with the residue retained, the addition of cover crops significantly increases microbial activities. The take home from these two slides is that the treatment interactions were different for each measure. And I suspect these interactions may vary annually, depending on things like precipitation and timing. But most importantly, we never observed negative interactions between the soil building management practices like residue retention and cover crops on the measured soil properties. So the rest of the presentation uses data from our long-term rotation study. This study was established in 2000 by our research agronomist, Shannon Osborne. All the plots are no-till. The corn and small grains are fertilized with 85% of the recommendations for our local yield goals. Treatment is crop rotational diversity. The default corn-soybean rotations were diversified by small grains, including oat, winter wheat, and spring wheat, also sunflower and field peas in combinations to assemble four-year rotations. Here we compare the effect of crop rotation on nitrous oxide emissions and soybean yields in the 2 and 4 year rotation. We compared the default corn soy to a corn, pea, winter wheat, soybean rotation, and we used all crops over the 4 years. I want to point out that all these treatments were replicated 4 times and that each crop was present in each year. And that meant for the nitrous oxide data that there was 32 plot years represented here and 64 for the 4 year rotation. For the soybean yield, each data point in the graph is from 16 plot years. And what you can see is that the 4 year rotation produced lower nitrous oxide emissions, and that means greater N retention in your fields, and that soybean yields were higher in the 4 year rotation compared to the 2 year rotation. These are the same plots, and we're looking at soil organic carbon. Soil organic carbon accrued significantly in the 4 year rotation, particularly in the 15 to 30 centimeter depth. That's about 6 to 12 inches. The corn soybean rotation did not significantly change its soil organic carbon over that 4 year period. Fortunately, probably due to the no-till situation. So here we've shown in these couple slides that the 4 year rotation not only produces greater yields, but it lowers loss of things like nitrogen, and it increases the gain in soil organic carbon. Now we are going to drill down and look at effects from sampling time, including which crop phase was present when the samples were collected, what the previous crop was, and what time of year these samples were collected. Again, we're going to look at erodible fraction. Here's the erodible fraction measured during the corn and soybean phases that are common among all these crop rotations. The erodible fraction was normalized to 100% for the corn-soybean rotation for both the corn and the soybean phase. Remember, erodible fraction, low is good, high is bad. Two of the rotations have lower erodible fractions in both the corn and the soybean. And those are highlighted with a brown circle. And you can see in both of those cases in the soybean phase, soybean was preceded by winter wheat. In contrast, in the other two 4-year rotations, there's a high erodible fraction in the soybean phase, and both of those, in that case, soybean was preceded by corn. But I also want to point out that erodible fraction in the corn phase is lowest in those latter two rotations, the one with the purple highlighting. And in those rotations, corn followed winter pea or sunflower compared to the brown ones where the corn followed soybean. Here's microbial activity for these same plots. Again, I've normalized it to 100% for the corn-soybean rotation. In this case, big is good, low is bad. It is the same treatment pattern, just reversed in direction because of the different measure. Activity in the soybeans following corn is lower than that following winter wheat. You can see that with those highlights. Brown, the soybean follows winter wheat, and purple, the soybean followed corn. So previous crop makes a difference. And also the ordering of crops in these 4-year rotations. One thing I've been thinking about is these samples were collected in June, so during the early growth stages of both corn and soybean. And I wondered if we would get a different result if we sampled after harvest in the fall. And so that's one of the things we're going to be looking at this year. Coming up is going to be some more data from these plots. We're going to look at how sampling time affects the results and how the type of measurement may affect your conclusions. We're looking at carbon mineralization, CO2 production. Similar to the Solvita field test, but with much different methods. We used flux chambers and went out there bi-weekly for the growing season, and I think a little less frequently during the non-growing season. And these are typical patterns that we see over the year. The patterns of CO2 production track annual temperature, and they react to periods of rain and drought. The two 4-year rotations produced a bit more CO2 than the 2-year rotation, but this could be due to either greater respiration by the plants or by the soil microbes. Here we're looking at extracellular soil enzyme activity called beta-glucosidase. This is the rate-limiting step to cellulose degradation. These activities were measured during the corn growing season in 4 different rotations over a 4-month period. And a few surprising findings. I was very surprised to find out that the activities in all of these treatments started high in the early spring and then declined during the warmest part of the year. The second thing that was surprising was how much effect the previous crop had. Previous crop, in this case, oat or sunflower, increased the amount of beta-glucosidase activity that you would find during this part of the season. The central part of the season from April through May. So if you were measuring soil microbial activity to differentiate soil health, when were your samples taken? And what crop phase did you sample? What was the previous crop? And what exactly did you measure? These questions are at the forefront of our research for the next 5 years. The remainder of this presentation involves these same plots and a recently published study demonstrating the influence of crop rotation and previous crop on soil microbes and crop performance. Here are the rotations that were studied: corn, soybean, corn, oat, winter wheat, soy, corn, pea, winter wheat, soy, corn, soy, spring wheat, pea, and corn, soy, spring wheat, sunflower. Again, each phase of the crop was present each year. In this study, we looked at 2 years, 2016, 2017. Looking at corn yield here in this plot on the lower part of the slide. The olive green bars are the corn grown in the corn soybean rotation. They had the lowest corn yield in both of these years. And so we set out to figure out why. Why was corn performing better in the diverse rotations? We sampled crop biomass, tissue nutrients, and we measured the soil rhizosphere communities. It's the microorganisms that are closely associated with the roots of the corn plants down to a depth of 15 centimeter. And we looked at both bacteria and fungi. We had competing hypotheses. First one is in the low diversity rotation, corn acquires fewer nutrients, which limits growth. The second hypothesis is that in the low diversity rotation, microbial communities suppress corn growth. We found that there was no evidence from soil or tissue nutrients to support nutrient limitation. So therefore, we focused our work on investigating the relationship of the soil microbial communities in the corn soybean rotation and how they might differ from the other 4-year rotations. We used a technique called structural equation modeling, and we found that crop rotation affects corn seedling biomass. And again, you can see that the olive green, this is the corn grown in the corn soybean, has the lowest seedling biomass, and seedling biomass predicts tasseling biomass, and tasseling biomass predicts corn yield. Here again are the crop rotations, the 2-year corn soy and the 4-year rotation. And we found that there's 2 types of rotational diversity effects on corn. One is the overall length of the rotation, whether it's 2 years or 4 years. The second is what were called the short-term effects. And this focuses on the previous crop, or the previous crop legacy is what we're calling it. And in that, we had 3 rotations that had soybean as the crop before corn. In the 2 rotations, we had pea or sunflower. Reminder that we're looking at crop biomass, tissue nutrients, and the root-associated microbial communities, both at the seedling and the tasseling phases. At tasseling, the corn plants are smaller in the 2-year rotation compared to the 4-year rotations. We saw this in both years, 2016 and 2017. So again, at tasseling, we examined the bacterial communities in the corn rhizosphere with DNA methods. And using something called factor analysis, we generated this x-axis in this plot, which reflects the essential differences in the corn root bacteria from the 4-year and the 2-year rotation. The bacterial communities on this x-axis on the left side are from the 4-year rotation, and on the right side are from the 2-year rotation. And you can see on the y-axis is corn biomass, that the microbial communities that were present in the 4-year rotation are associated with higher corn biomass at tasseling stage, and in the 2-year rotation, lower amounts of corn biomass at the tasseling stage. We also found that the biomass of corn seedlings was smaller following soybean compared to following pea or sunflowers. And this held true for both years that we performed this study. We examined the corn root fungal community by DNA analysis and projected on the x-axis the essential differences in the fungal communities following either soybean on your right or peas and sunflowers on the left. The fungal communities colonizing corn roots following soybean were associated with lower seedling biomass, which we have shown earlier predicts yield. In contrast, the fungal communities associated with the corn following pea or sunflower were associated with higher amounts of biomass. And this is where our research stands as we follow up on these relationships and try and understand the mechanisms behind the relationships between the prior crop, the microbes, and the performance of the corn. We certainly don't know all of the details, and there are a lot of interactions in these complex systems. But it does seem clear that stacking as many positive management practices as you can within the constraints of your own system will increase the probability of the emergence of beneficial properties such as yield, resilience, increased soil carbon, and conservative nutrient cycles, in addition to increases in soil structure that we observed by measurements of erodible fraction. And with that, I'd like to close by thanking you for your interest and attention, and to acknowledge my great collaborators, Shannon Osborne, a series of postdoctoral research associates, and a lot of technical help from students, and Sharon Nichols, my technician. And I would like to entertain any questions. Thank you.
Did you record soil temperature and moisture at the seedling time?
For which study are we talking about?
For the seedling biomass.
Oh, the seedling biomass.
Yeah, did you record soil temp and moisture at that time period?
We do have soil temperature and moisture. Those are kind of routine measurements in a lot of our plots.
were they the same?
Were they the same, or was there some differences that you recall?
Oh, no, you mean same in terms of the years or the treatments?
The treatments as far as when you're looking at the seedling biomass.
Oh, you know, that's something I'm really interested in, and so far we haven't seen differences in that kind of data. And we have another field experiment where we have about 5 years, I think, of temperature data that one of our workers is going through, and I'm very interested in that correlation between the soil temperature and moisture and what the microbes are doing. That's definitely going to be a bigger part of our research program here. But we do have spot measurements for all those plots for soil temperature and water, and I don't believe there was any differences between the treatments. Of course, the years were totally different. I mean, the weather and moisture in 2016 and 17 were much different from one another.
Okay, anyone else? Yeah, I think the biggest thing is that ground warmer under like sunflower stalks, and is that why you had more seedling growth there versus on soybeans?
Did you hear that, Mike?
Not the whole thing.
Was it warmer under sunflower stalks versus soybeans for the seedling growth?
I'm thinking about that.
I mean, we can make assumptions, but that would definitely help in research.
No, I think the answer is no on that, but I don't have the data right in front of me. All those sunflowers were harvested, the year before, right?
Yeah.
So I don't know if the staff would make that much difference, but that's something else we can look at.
Okay.
Okay, thank you. Anyone else? Okay, thank you, Mike. We appreciate it. Hey, let's just take. Oh, we got one.
I just wondered if the tillage system was the same across all rotations.
The tillage system is the same across all rotations. It was all no-till, right?
It's all no-till.
Okay, thank you, Mike. Appreciate it.
Okay, we guys have fun.
Okay, thank you.